So I want to thank you for attending once more. As everybody knows, this is the virtual version of INTERPHEX Live. Customarily, we come to you hot off the press here at the Crystal, not here, but at the Crystal Palace in New York City at the Javits Center. Well, we won't go into why we're not at the Javits Center. So, but one of the panel topic today, with the help of Katy, has progressed over several years. Those who've been with us and follow the discussions, we started out a very specific by calling it we called it the CAR-T panel. And as you can see, we've moved on beyond, and we start to talk about the whole landscape, which is the key today. And having said that, I want to introduce our speakers. We have Diane Blumenthal, Head of Technical Operations at Spark Therapeutics. Norman Goldschmidt, as always, Norm, hi, welcome, President of Genesis AEC. Audrey Greenberg, Executive Managing Director at the Discovery Labs, welcome. And my friend, Katy Worrilow, CEO of LifeAire Systems and an associate of mine. And I also like to introduce Amos, Principal Scientist at Janssen Research and Development. So based on that, what we're going to do is we're going to start off and we're going to put some context around, given that the rapid growth in the cell and gene therapy technologies, it may be, I think what we need to have our panel provide a brief definition so we can get some context around what we're going to be talking about today. So Amos, so if you don't mind, I'll turn that over to you.
Thank you, Russ. Just want to introduce myself, Amos from Johnson & Johnson. Before we start, I just want to kind of level set our expectation about what we're about to discuss today. This is a very exciting technology that we're dealing with in terms of cell and gene therapy. The exciting science is happening at a very, very rapid pace, and compared to what we have observed and been able to accomplish with other biologics that we've been able to successfully commercialize in the last 20 and 25 years or so. As such, I believe that there are a lot of things that we can still learn from our past experience in other biological modalities that would help us better position ourselves for success in cell and gene therapy. There are some commonality that I see with respect to our historical knowledge. However, there are also a few things that are unique to this particular platform that we should also be aware of. So I would just like to start off in sharing with you what I think are likely to be quite similar to what we've known from what we've learned from the existing biological platforms. For instance, the process through which we are making our drug product, whether it's cell or gene therapy, I believe that we're still relying on the same sort of a physiochemical characteristics to help us manufacture these drug products. So therefore, the unit operations would tend to look quite similar, or at least reminiscent of what we've been doing in, for instance, the monoclonal antibody world. And so therefore, we should be able to leverage a lot of learning from that space. In addition, we have been rapidly automating and intensifying the MAP processes to reduce costs, to improve our quality of the product, and have better control over our overall process and operations. I believe that is something that would continue to be significant and relevant to the cell and gene therapy world. Therefore, it is something that is worth considering at the start, rather than trying to play catch up 20 years later, like what we're doing now in the MAP world. In addition to that, the perennial question about whether we should build one's own facility to be ready to commercialize versus working with the CMO community to help us cross the finish line. Those conversations remain relevant just because the business landscape remains the same, that for a lot of startup company, we're faced with limited capital, for instance, versus the ability to actually maintain better control over your commercialization timeline. So those things remain relevant and similar to what we have, to the knowledge that we've accumulated over the last 20-25 years. What we see could likely be different is that the CMC life cycle is very, very accelerated in a cell and gene therapy world. Therefore, as such, it is highly, highly important that we try to figure out how to minimize the number of process changes that we will have to perform prior to commercialization, and try to achieve the idea of single cycle development as much as possible. That is, I believe that's something that is becoming rather critical. In addition to that, we believe that the cost structure, component distribution might be a little different in terms of the idea that raw material cost seems to be now occupying a rather bigger part of the pie. And that's something that is largely due to the fact that there are new technology that we're dealing with, such as vector delivery and for gene therapy and gene editing, and also these personalized medicine requirement that we need to meet. Therefore, it really adds to the cost of raw material that is representing a larger part of the total cost that we would need to somehow figure out how to mitigate. And with that, I will just hand the discussion to my colleague Diane and have her to share her expertise and experience in the cell and gene therapy world.
So thank you very much, Amos. So to follow on with your discussion, I'd like to talk a little bit about cell and gene therapy from the perspective of some history around how technologies in the biotech arena have come about, and some commonalities that I see. And we'll talk today amongst the panel about cell and gene therapy as a collective, but we will also try to gear our conversation and be specific around things that apply and discussion points that apply more to gene and cell therapy, more to gene therapy and gene editing types of technology, because those are uniquely different than cell therapy based technology. And what I mean by that is that the gene therapy and gene editing types of technology are more geared towards typical types of biotech processes, and we'll talk a little bit more about that in the panel. You are delivering genetic material to specific types of cells, and either correcting the genetic deficiency in those cases of gene editing, but certainly in gene therapy, we are delivering copies of the gene to the cells so that the correction can be made, at the cell point. And we're not necessarily in gene therapy correcting the genetic modifications or mutations, but more providing a correct piece of the gene. Cell therapy is quite different because that's more personalized medicine. And the manufacturing and CMC considerations in a cell therapy situation can be quite different. So today, as we're speaking, to the best of our ability, we will try to make distinctions between those two types of therapies, because they are different, certainly from a CMC perspective. And then just briefly, so we can move into the questions, if you look back in history and you look back how many technologies have come to bear to patients and then certainly been perfected over time, these patterns are somewhat cyclical. Researchers go into the lab, they identify new modalities, and then we try to bring those to patients. And in the early parts of those life cycles, some of the manufacturing processes are, a bit more research-based. And then we as CMC professionals need to take those and put good engineering principles to them, and take some of the things that we've learned from past history and apply those. And that is very much the life cycle of what we've seen, even if as you go all the way back into antibiotic technology and penicillin technology, the way we made antibiotics and penicillins, certainly the way we made drug therapies in the early days before recombinant DNA was popular, was much more laboratory based, much more labor intensive. We learned how to do submerged tank cultures. We learned how to make antibiotics and certainly penicillin in very, very large quantities. When we moved into the recombinant DNA technology world, and even the mammalian cell technology world, it took us some time to get those engineering practices put into place. We have been very successful in a very, very quick period of time to take gene therapy technology and cell therapy technology and advance it very, very quickly. And in the 5 years that I have been at Spark, I have seen a major, major change in how we are delivering medicines to patients. And I'm very, very excited about how we will be able to advance our technology and grow it in a much more rapid pace than maybe we did, over the decades in more mainstream biotech and monoclonal antibodies, and even in the early days of protein recombinant technology. So I'll stop there. And I'll pass it back to Russ as our moderator, and he can start to ask us some of the questions that we have prepared for you.
Thanks, Diane. So there's the background. And one of the commonalities, myself, just to give you a background, I've worked in late stage development, so we touched on a lot of stuff, none the least of which is manufacturing and getting it to the patient, which brings up ever popular tech transfer. So, Audrey, we would, as we were talking about technology transfer of the gene and cell therapies. Obviously, a lot of this or some of this differs from mainstream biopharma, and manufacturing certainly differs from mainstream small molecule. And so these unique needs. What are you guys are doing, and how are you addressing those needs?
Yeah. Thanks, Russ, and hello, everyone. Obviously, with cell and gene therapy, you cannot get closer to the patient when you're dealing with these modalities, and the process, as we all know, is the product. Technology transfer is more important in this particular arena than in others due to the complexities involved with the manufacturing process. And depending on where a product is in its life cycle, it can be more important than ever. In particular, with small companies that may have developed a technology in a lab that was not really contemplating large-scale production, and rather these companies are very focused on innovation and maybe not incorporating the big picture of commercial production. In addition, I'll add, these companies are very incented to by speed to market in capturing the patient, and may not necessarily be considering the downstream impacts of the decisions that are made early. So at a CDMO, like the Discovery Labs, where process development and tech transfer is really the gating item for these therapies, you're having to deal with every product being very much customized and different. There's not a tremendous amount of synergy across platforms to the extent that these are very much, like I said, customized. With cell and gene therapy, rather than traditional pharma, we're really in our infancy relative to, mAbs and some of these other products. And these medications, like I said, are going through the process of milestones, right? So these companies are funded by private equity firms. They have milestones that they need to achieve based on phases of their life cycle and accomplishments that they're going through. And to the extent they can quickly go through these processes, they get funding much more quickly. They have speed to market. But then they come to the CDMO and they say, help me mass produce. And we're maybe stuck with processes that aren't that scalable. So we're dealing with some of these issues. However, given that we've done this before with other therapies, even though that each process is unique, we are able to implement best practices in terms of upstream, downstream, purification and the like in terms of suspension versus adherent. And to the extent companies are not making major changes, you can still streamline the process with the FDA. And then I'll say, obviously, making something scalable, repeatable, and keeping your quality in line. All of those things are very much an important part of the tech transfer process.
Thanks, Audrey. I mean, but you know, one of the things that we've talked about a couple of times, and I think a lot of the stuff that happens, at least in my experience, has been, we talk about tech transfer, but the landing pad, as it turns out, when we get there, it's an, oh my God, you know, there's things that we don't anticipate. And I have to say, my experience has been, you know, especially in this area, as limited as it is now for me, is it's always the things you don't expect.
True.
Thanks to Katy, I'm gonna ask her the question, which was a revelation. And Katy, just so you know, it was a revelation again last year for about, as you remember, we must have had 15 people line up to question things, and that is, from your experience with live cells, what are the challenges that we don't even anticipate when it comes to the cell and gene therapy? And where's the unknowns that are hurting?
Well, thank you, Russ. And good morning, everyone. First and foremost, I'm a cell physiologist and was a clinical provider of human in vitro fertilization. So our whole world focused on the culture, the successful culture of the human embryo for up to 7 days. And we've really spent the last 25 plus years studying the molecular epidemiology, if you will, or the molecular role of specific airborne metrics in our in vitro culture environment. So, you know, you don't know what you don't know, and we've learned quite a bit about the specific and combined role of particular airborne metrics that are not controlled by your traditional clean room. We actually performed IVF for years in an ISO 5 clean room, and what we learned through our own cellular failures was that the clean room does a phenomenal job of controlling for your viable and your non-viable particulates. But what was missed and what we learned was the control of your volatile organic compounds or your chemical pathogens, very common to really all cell culture laboratories, manufacturing facilities. And we're starting to see the presence of these very common and frankly high levels of VOCs in some of these cell therapy manufacturing facilities. And so to answer your question, there are a lot of new known variables which are negative and significantly impactful on successful cell growth and cell viability, cell slow, cell arrest, cell loss, and we experienced that ourselves until we identified what level of overall VOCs were paramount for not only our successful cell growth, but consistent cell growth. And we also learned what was significant in our outside source air that is common that you really can't control, you know, whether it be resurfacing a road or resurfacing a rooftop or construction. Biogenic VOCs are very common, why a lot of people see seasonal variability in their cell success. Shrubs and trees. Also, those VOCs that we generate as staff, the materials that we bring in, our SOPs, our protocols, things that you can't avoid. It's just a necessary part of the process. So the new known negative, and we've had a lot of discussions with cell therapy and CAR T manufacturing facilities in completely remediating and removing this variable from the process. There are a lot of variables, but this is one that is extremely impactful to the living cell. Just one other factor here, we've also learned how VOCs impact cytotoxicity. So if you have 100 parts per billion IPA in the air, which everyone uses, how long does it take that IPA, those molecules, to enter your cell culture media, even in a closed system? And then how long does it take to go from the culture media to the living cell? And once present in the cell, exactly how does it exert its toxicity? So there's impact on DNA replication, RNA replication, DNA methylation, gene expression, and this is why it has such significant impact on your cell culture metrics.
So, you know, this always amazes me. So one of the things is that when we talk about capacity, we talk about location, and these other things come into play. So, Audrey, there's got to be a lot of constraints around increasing manufacturing capacity. Where are the stumbling blocks or barriers that you run into?
Yeah, we're all very familiar with the cost associated with building a biopharma facility, upwards of $1,200, sometimes $2,400 a square foot. So cost is a huge issue. That's first. Second, time. You've got to find a location. You've got to get zoning, which can be very, very challenging. You've got to design, and there's a lot of competing priorities and constituents that want to raise their hand and offer their opinion. And so time, it can take up to 2 to 3 years to get a site online, and that's fast tracking. So cost, time, talent. We're all lucky, many of us on this call today and this video, to be located in Silicon Valley in Philadelphia, where there is a surplus, 50 to 60,000 life science experts in our area. So we're lucky to be based here, and it's easy to procure talent in this location. But there are locations where that's not the case. It may be inexpensive to build there, or it may be, in the case of a urban core, where you want to be located close to academia in some of these urban cores, and it's very hard to build horizontally in those locations and to get zoning. So there's a lot of considerations: cost, timing, talent, and really, interestingly enough, the equipment and consumables are some of the longest lead time things out there. Talking about some of these bioreactors, even bioreactor bags, it can take longer to get than maybe even hiring a chief scientific officer. So there's a lot of competing things. We're lucky in that, like I said, we're all located here, and the Discovery Labs, our CDMO, we're bringing on 616,000 square feet of manufacturing space. So we have capacity coming online. We've got the talent. We're lining up the raw materials, the consumables, and all of that wonderful stuff. Lastly, I'll just say, what could possibly be impacting some of the capacity constraints, really, and what we're experiencing in terms of having to wait up to 18 months for a CDMO, and 3 years to build your own facility. Really, the process is a very important part of that. To the extent that you're scaling out instead of scaling up, you're going to need a tremendous amount of real estate. It's very real estate intensive. So coming up with better technologies for growth, maybe even non-viral means, things that create a process with a smaller footprint, will provide for easier accessibility to space and throughput.
So those are the solutions, some of the solutions for these.
I would say those are some of the solutions. Supply chain is an important part of it. I mean, once you have your facility up and running, and we all know that can take some time, or once you've entered into a relationship with a CDMO, supply chain becomes a very important part of getting patients the cures they need. So you've got your capacity, but then what do you do with it? All of a sudden, you're out of cell stacks, or you can't get a bioreactor, you can't get a bioreactor bag, you can't order those things on Amazon. So we're faced with supply chain disruptions, even outside of the pandemic, which is currently occurring and making things even worse. We're really faced with things in supply chain and technology, whether it's on the upstream side in terms of cell growth, whether it's on the downstream side in terms of purification. And then in cell processing, isolators that will provide for ballroom type setups and lower grade space that is obviously easier to build and more quickly come online in terms of air quality. So all of the things in terms of technology that can enhance our end-to-end process and supply chain, which can make the process more efficient such that there's not bottlenecks when you're going through commercialization and manufacturing, those things will all make a tremendous impact on the efficient delivery of therapeutics to patients.
Well, when we talk about supply chain and we talk about raw materials, Audrey, I think you bring up a couple of great points. But I think the thing that always catches me flat-footed, and I admire my colleagues in the cell and gene therapy area is that, you guys deal with patient cells, which is essentially the starting material. And I'm always like, amazed. So Katy, there got to be some unique considerations when you're talking about patient cells, and that's the starting material. Can we touch on that a little bit and talk there?
Absolutely. Again, I'm very cell-centric, but whether it's the human embryo in our case or the cells cultured in cell therapy or CAR T, they all carry an exceptionally high therapeutic value. And in the lab or in the manufacturing facility, we have one chance with these cells, one chance to successfully culture them and deliver, hopefully, a successful cellular product to the patient. So in addition to their high therapeutic value, they can come from a varying degrees of compromise from the patient environment, depending upon the illness, and therefore are extremely sensitive and extremely variable. So your starting point is difficult. And so given their sensitivity, again, I can't emphasize enough, and honestly, just coming from our own learnings over the last several decades, the significance of the environment outside of the typical parameters considered for the clean room, in particular, your chemical and VOC levels. In addition to those, you need to carefully look at the fabric of the space, if you will, the walls, the ceilings, and Norm probably knows this better than anyone, the components of that fabric of the laboratory, the gasketing, even new equipment can continue to off-gas, new gasketing, new linings, tubing, and can be somewhat of a perpetual source of cytotoxic VOCs, but also looking at the engineering design, the airflow crosstalk, if you will, between the different spaces where the different activities are occurring, and just, again, just negating those now known cytotoxic variables in that environment, given the sensitivity of these patient cells.
I think, how can I put it differently? We talk a lot about the challenges, but, so Diane, at Sparks, so what are you guys doing to increase your speed to market? Can we talk? There is incredible amount of opportunity. So, we've heard about all the stumbling blocks. But, so, and you got to be under a lot of pressure to get. So, where's the path? How do you get new products to market and fast?
Yes, that's a very good question, Russ. And so I'll address that question, that they're different for the gene therapy area than they are for the cell therapy area. And while I'm not an expert in cell therapy, I can make a few points, but let's start in gene therapy first. So gene therapy, the good news is that we are moving now more towards traditional processes in gene therapy. And what we're moving towards is taking a lot of what we've learned in the last few decades in process development and certainly manufacturing efficiency, and applying it to the processes that we use in gene therapy. And the goal here is to come up with several standard processes. In best case scenario, it might be even one standard process, although that's not necessarily always feasible. But to try to streamline things to make them as platform as possible, so that each time we're developing a new molecule or a new transgene, and we know what cell type we're going to deliver to, we know which capsid category we want to focus on, because the capsids are specially engineered to deliver to certain types of cells. And then we pick the transgene that we need, and then the process then becomes sort of the building blocks of each of those different things. And then we have standard practices on how we develop these and how we move them through to commercialization. And we're not reinventing the wheel every single time. And so that's where we're moving towards. Again, building blocks of platform processes. The other piece that's very important, and this applies to both gene and cell therapy, which is understanding our product better. Biotech has been around for a long time, recombinant DNA technology and proteins, but even more importantly, monoclonals. You sit and watch TV in the evening, and monoclonals are used for a wide variety of things now because we've been working in this area and developing them for many decades. And we understand them from a molecular perspective. So whenever we need to make process changes, facility changes, scale changes in those arenas, it's not the hurdle of getting through the comparability exercises is fairly streamlined now. We need to get cell and gene therapy there. In gene therapy, we're making great strides to being able to understand analytically what we have, understanding structure function, understanding impurities, and trying to minimize those impurities and improving transfection efficiencies so that we are getting a purer product where you don't have, a mixture of empties and fulls and then partially fulls. And the transfection efficiency is very, very dependent upon the biology, and we need to improve that biology. So we're, again, taking, learnings that we have had over decades in analytical science, in process science, and certainly engineering science to bring these products to patients. Even biological science, because the transient transfection process is the part of this that is not as scalable as we would like it to be. Growing mammalian cells we've been doing for a long time. It's the transfection process. So moving to packaging cell lines, moving to producer cell lines. In the area of cell therapy, I'll just touch on it very briefly because I'm not an expert in this area. But again, engineering practices, learning how to, maybe bring the manufacturing factory, so to speak, to the patient bedside. Decreasing the probability of having challenges with patient cells having to move through the transportation system to the site of manufacture, the time it takes to do that, then the time it takes to process. And I do see a lot of companies coming out with some very, very interesting technology. And regulators are pushing companies to really try to move a bit closer to the bedside. Of course, that brings with it GMP challenges, training challenges, aseptic challenges. But we have a lot of very smart people out there who have a lot of very good ideas. And I see a big shift in how cell therapy processing is moving forward just in the last 5 years since I've been working in this space. So I'll pause there and turn it back to you, Russ.
Well, thanks.
Yeah, Russ, if I may just kind of second Diane's point around the criticality of understanding our product better. That is extremely critical in our attempt to accelerate our timeline, because we need to know almost in real time the impact of our CMC action on the product, right? In addition to, during the manufacturing process, what, if anything, we're doing that adversely impacting our product. So the need to kind of enhance product understanding, product characterization. But from a technology development perspective, more importantly, is bringing online, these characterization methods in a high throughput at line measurement format would be extremely valuable for our collective effort in accelerating development timeline.
So Amos, coming from a large innovator company like Janssen, so let me ask you this. I mean, how do you determine facility design and profitability for commercialization? And what are your options? And, in other words, we hear about scaling out as opposed to scaling up. And I always find that discussion interesting, because in small molecule, we don't scale out. It's like a new concept to us. But anyway, right. Scale up versus, what are you doing? How do you assure commercialization and profitability?
So just because we're a big pharma doesn't mean that we are any smarter than anybody else. We are working under the same constraints. The constraint that imposed on us by biology, by engineering principle, just the same as everybody else. The only difference that I can see that we have some advantage over is that because we have experience in other biological modality, as well as on our enterprise level, we have other business in place that would give us a better understanding of how we can actually improve our efficiency. So in this case, I really don't think that the difference is really around between scaling out and scaling up. as long as the scaling is done linearly, we're not really buying any efficiency, whether you're scaling out or scaling up, right? This is why, you know, we have this derogatory term to describe biopharmaceutical plant as tank farms, because that is a result of linear scaling, even though we're scaling up. And these facilities are humongous. They take up a lot of land, even though it's out in the middle of nowhere. So we believe strongly that linear scaling, whether it's out or up, is not really the appropriate approach for this personalized medicine space. But the idea of process intensification, process automation, connecting processes in a closed manner, that probably is where we would realize improvement in efficiency. Only through improvement in efficiency will we realize improvement in cost. So that's an area that we would develop and deploy a lot of the resources into, and also leverage our internal experience across our enterprise to see, like, we can actually surprisingly learn from, you know, our Listerine colleague about how automation could improve our processes, for instance.
So yeah, you bring up a couple of good points, and so you know, if we have an, so let me ask the question of Norm this time. So I have an existing system and an infrastructure, so I want to get into the CAR-T business, the cell and gene. So I mean, what do I need to do that's new? What do I need to do to get into the game? I mean, if I don't own a tank farm, I don't sit in the middle of nowhere. And so far, I've heard a lot of it, but let me hear the other side.
Sure. So there's a lot to unpack there, Russ. And there's been a lot of very deep discussion so far on, quite frankly, the difference. We talk about cell and gene therapy as if these are somehow the same. And of course, they're very different technologies, as Diane said, right? So let's separate what we know, what we're good at, from those things that are really cutting edge, and what has to be different. We can make buffers, we can make media, we know how to make aseptic solutions. Those parts of the support process we're good with. In the gene therapy space, a lot of our toolkit from doing therapeutic proteins is quite applicable. So we're doing chromatography, MFUF, a lot of the technologies that we have developed and really gotten good at do apply. The real outlier, I think, is in the cell therapy space. So you said CAR-T, right? And with cell therapies, we're a bit back at the beginning. I feel like it's early in my career again, back in the 90s when we were doing you know, our first biologics plants. And Audrey had said, right, this is the product is the process. And that idea that all of the variability that's associated with the individual cells that you're dealing with has to be dealt with, and somehow we're supposed to get a reliable, repeatable process out of very different starting materials. That's the challenge. We have cell therapy products can't be sterile filtered, so they're aseptic much earlier on. So for a lot of these cell therapy processes, we're doing aseptic technique and aseptic facilities very early on, very highly classified, which drives back to what Amos said about intensification. The more we can take those processes, which are so sensitive to environment that Katy's been hitting on, the more we get them closed, the more we can control the environment that they see, the better we can intensify and not have huge build-out footprints. So we're really on the cusp here. It's a very exciting time. In the cell therapy space, it's about getting it closed or getting it very highly classified, getting, as we have more automation and intensification and closure, allows us to shrink those facilities down, because scaling out, as Audrey said earlier, just takes a huge square footage. And it becomes such a bar to entry for some of these really leading edge therapies. On the gene therapy side, The challenges tend to be more the kinds of things that we saw with vaccine facilities. So there we have, about cross-contamination. It's about the risks associated with having the vector present, purification, aerosolization of the vectors, and that's where a lot of what we know about control of the environment can really help us to decrease the, and I hate to use the word vector 2 different ways, but decrease the vectors for cross-contamination so that those very individual products can be kept separate. So the facilities, try to pack this back up again. The facilities are different because they tend to be much more highly classified earlier on. We have much more risk of cross-contamination, and unless we drive towards intensification, they're also a lot bigger. Back to you.
You bring up a lot of great points, Norm. I mean, if I was sitting there trying to decide what to do, you'd be giving me a headache. But anyway, so we've heard that it depends on the community you're in. It depends on the air you're breathing, as Katy says. So Diane, let me ask you the punchline here. So we're trying to reel this one in. So, as you know, people that we work for ask very simple questions. So let me ask you the simple question: do I buy or do I build the facility?
Oh, so yeah, that's a question we ask very often. I've been through this at many companies that I've worked at, and it's a complicated question. It's certainly for Spark. Back when Spark made the decision to move Luxturna forward in the regulatory process, there was no contract manufacturing. That process had been developed many years before by the folks at Children's Hospital who had acquired that technology from a small biotech company out on the West Coast. Much of the clinical material was made at the hospital where the clinical trials were being executed. And so there was no contract manufacturing. It was either continue to manufacture at Children's Hospital, and I know Norm knows this very well, or continue or build their own manufacturing. And so that was a very easy decision for Spark, excuse me, because there was nothing else out there. Even the whole concept of fill finish and doing our own fill finish was a little bit of a challenge because that's a very specialized skill set. And we would have kept that in-house if we didn't have options out of house. But as the technology has developed and as companies have gotten on board, there were quite a number of very, very good CDMOs that are out there. And so for a company that's just starting in this, who doesn't have a lot of capital, it's a big lift to see them build their own manufacturing facilities when they don't really know whether their product will be successful or not. And what happens with many of these startup companies, as I've experienced, is you have your first product and then it takes a while before your next products, come down the pipeline. So, CDMO is a good choice for the beginning until you get an infrastructure in place, you have the right staff, and you're really ready to make that investment. For more established companies, the larger pharmas like Johnson & Johnson and Pfizer's, and certainly, as everybody knows, we're now owned by the Roche Group. There is the ability to have that capital infrastructure and to do it sooner and have your own facility sooner rather than later. Despite how they are all the CDMOs out there, certainly the ones who have gotten into this business early are very, very capable. There is nothing like doing your own and having control over your own. And there are solutions out there to getting dedicated suites at some of these companies so they can be satellites to your own company. But it is a tough decision, and a lot of it is driven really mainly by capital as well as know-how. We keep all of our analytical in-house because that is, where we feel that there's a proprietary advantage to keeping that in-house. And that is the approach that Spark has taken. So a mixture of in-house manufacturing, external, where we didn't have the scale that we needed, and then we keep most of our analytical in-house.
I got you. Norm, you had something you wanted to add?
Yeah, I really wanted to reinforce that last point that Diane made, because so often yes, it's a capital question. It's certainly depends on how you're funded, but it's also a talent question. So at different companies in different positions, if you simply don't have the resource, and Diane and I went through this, where we took a process that was coming out of a hospital lab, which had process steps in it that we couldn't commercialize. Getting somebody like a CDMO, if you don't have the staff to help you make that transition from a pure lab process to a commercializable process that's scalable, then that's a great thing to, right? That's a reason to lean on a CDMO, because you simply don't have the resources to help you make the transition from early lab, maybe developed in a hospital or some other research institution, to commercializable the way classically a big pharma company would be able to do through their tech transfer process.
I understand. Again, Audrey and I talked about, we're going to move on to, I think the punchline is coming up, but Audrey and I spoke about this the other day, and to work with the CDMO, the assumption is it's not, you can't throw it over the fence. That's a, buy or build, but the answer is you got to work closely, and you have to have that agreement. Audrey, I mean, that's got to be key here, is the agreement and the working arrangement. I mean, you're not, you know what I'm saying? You can't give them all the answers, right?
Yeah, that's right. I mean, this is all about a partnership and working together. Life science and cell and gene therapy in particular, across all industries that I've worked in, is the prime example of trust between counterparties, and relationship is incredibly important. These are long-term relationships where lives are at stake, and it's an incredibly important relationship that requires incredible amount of transparency and communication and reporting that I haven't seen in any other position that I've had. I would say that. And then the other thing I'd say, just piggybacking on Diane and Norm's comments, is that there is this concept of redundancy with the FDA and for compliance reasons. So yes, we all understand that for small companies that are capital constrained and need efficient use of cash, really outsourcing is maybe the answer, and the extent that they are adhered to a certain process, which may or may not be scalable. Certainly you want to incorporate a CDMO such that they don't have resources to do so. But then I'd also like to say that even for large companies with very scalable processes, it's important to have redundancy with regard to facilities in case of what we're experiencing right now. So pandemic or otherwise, where you really want to have a backup plan. And just commenting also that what we're seeing out there in terms of demand from the customer side, in addition to these smaller companies that want process intensification, process development, tech transfer, etc., we do see very large drop-in clients from big pharma that, yes, they want to control their process, as Diane was talking about, but they also can't get capacity. And so unfortunately, that is at play right now, where capacity, we've got 1,100 in development with the FDA, and the FDA pipeline, there's 10 to 20 projected by Peter Marks and the FDA that are going to be approved each year. Any single one of the large indication that require a lot of vector could take up a tremendous amount of capacity. So that comes at play as well.
And just, I want to bring this back to the discussion that Amos and Diane were having too. I mean, that was something we wanted to touch on with the CDMO. We'll come back to that if we have some more time. But Amos, Diane, I think one of the key pieces here is we talk about capacity, but let's talk about cost of goods. That's, I mean, big or small, it's, it's still the cost. So let's touch on that. Where are we at?
So maybe I'll start off based on to what I know of cell therapy at the moment, and also just my knowledge about, compare that and contrast it against the map world. Just because we're talking about a large company, we're still we're still under cost constraint, just like everybody else. It's just the calculus is slightly different. So in terms of COGS, certainly owning a manufacturing facility is part of the overall COGS versus buying capacity. So that is part of the calculus that is continuous, that never stops. that goes on through the entire life cycle of the product. For cell therapy, at the moment, if I look at the pie chart, the usual suspects remain the same. Is really operating a facility internally, if you have one, and the operating cost, really a large part of it has to do with the manpower that you need to staff a facility, which is a major cost driver, just like a typical monoclonal antibody plant. In addition to that, what we're seeing now is slightly different in cell therapy, is that the raw material cost is, as I said earlier in the introduction part of the meeting today, is showing up as a higher percentage of the pie chart that we are trying to mitigate through better biology. Because we don't believe that engineering solution will make a meaningful difference in the raw material cost, because a lot of this has to do with the inherent biology that we need to execute. Besides that, really is the continuum of cost reduction, little bit by little bit by little bit, right? To do kind of a routine debottlenecking exercise so that we can get another 5% here, another 10% there. So for instance, right now by default we send everything out in these liquid nitrogen door. Is there another way for us to do that, to kind of cut another certain percentage of that cost, in managing our cold chain, for instance. So the way we're dealing with it now is continuous improvement through debottlenecking. Once you debottleneck the first one, you kind of do it stepwise, in a stepwise manner. Take the biggest cost driver and see if you can do anything about it. Just work it down little by little bit. At the moment, really is manpower and raw material.
Well, we're coming down to the wire here, Diane, but I really want to hear. I really want to hear your spin on the cost of goods.
Yes, thank you, Russ, and thanks for that very nice introduction to and your thoughts, Amos. I think for me, it comes down to a couple of basic principles. First of all, your raw materials. In these processes, plasmids are very expensive. And as we scale these processes, they are a very large part of the cost of the raw materials. And so, as we can move away from triple transfection to, packaging cell lines and then ultimately producer cell lines, we can take a very large element out of the cost. The second thing is you don't really get the cost of scale. We all know that a large part of the cost of, manufacture is each batch, the QC testing of the batch, the QA evaluation of the batch, and we're not getting economy of scale here. Cell therapy, it's one process, one patient, one process, one batch, and even in the gene therapy space, we're not talking about large volumes, and because the amount of material we need to give to patients is very small, but even more so because the patient populations for these rare genetic disorders are small, and so again, you are not getting economy of scale. And then it is a one and done therapy. And so, you're not having repetition of patients coming back and needing, the therapy, as an ongoing therapy. At least that's the way we see it now. Maybe in the future there might need to be redosing, but we're a long way away from that. So, it's those three categories. And so we're not able to get the economy of scale, the efficiency of some of the things that are the high cost elements. And scaling out is costly because of land and manpower. And then as we've moved to the third category for me is as we've moved away from highly automated facilities, stainless steel facilities, and there's reasons that we've moved away from them, and we've moved to a lot of single use. Single use has its advantages to speed in bringing facilities online and changeover, but we're throwing away things every batch. And I do think that we will start to move a little bit back to the middle, see some single use, some reuse, and we need to be a bit more efficient in how we're using these materials. Chromatography resins are expensive, membranes are expensive, and if you're throwing these out with every batch, it gets to be costly after a while.
Thanks, Diane. So we have come about almost to the end of our allotted time. I want to thank everybody. I mean, we didn't even have an opportunity to talk about current supply chain constraints. However, I think that becomes obvious. I think Diane brought up a couple of things that we're going to look at for live sessions in 2021, none the least of which will be the fill and finish aspect and dimension, which we, for some reason, we never address it here. So having said that, I want to thank everybody for their time, and hopefully we will see you at the Javits Center next year. Thank you very much.